Welcome to the deep dive. Okay, so today we're kind of putting away the genome editing scissors. We're
Yeah.
moving beyond just, cutting and deleting DNA. Our mission today is a deep dive into CRISPR activation. CRISPR, they call it.
Yep, CRISPR.
We're talking about precision genetic programming. So the ability to basically flip a switch and turn our own genes on exactly when and where we need them.
It's a really exciting area. I mean, this tech is already being looked at for some incredible things, regenerative medicine, even fighting stubborn cancers.
Yeah, it feels like a fundamental shift. When people hear CRISPR, they almost always think Cas9, the molecular scissors,
Yes,
right?
that's the common image, yeah. But CRISPR, it really changes the game. Instead of cutting or trying to fix something, we're focused on robust gene induction. We're basically telling the cell, hey, make more of this protein you already know how to make.
Okay, let's get into the tool itself then. The core component is a modified version of Cas9, deactivated Cas9 or dCas9.
That's the one.
So for listeners, how exactly do you take this really powerful cutting enzyme and, well, turn it into more of a GPS device, something that can find the spot but not cut?
Yeah, it's actually quite elegant. Researchers introduce very specific mutations. The sources mention D10A and H840A specifically.
Okay.
These go right into the critical parts of Cas9 that normally do the cutting, the nucleus domains. These two little changes essentially defang the protein.
Defang it, I like that.
It keeps its amazing ability to be guided by an RNA molecule to a super precise spot on the DNA. Unmatched precision. But it just completely lacks the enzymatic power to make the cut. It becomes this perfect programmable scout.
It's pretty wild that just two tiny changes can totally flip the function of such a big protein.
It really is.
So, okay, now you've got this perfect DNA binding scout.
Yeah.
But just having dCas9 sit on the DNA, that doesn't automatically turn a gene on, does it? That's where the activation part comes in.
Precisely. Yeah. Binding alone isn't enough. To actually flip that gene switch, dCas9 is fused, like physically linked to powerful activation domains. Waggies.
Activation domains.
Yeah. Yeah.
Think of these ADs as protein recruitment magnets. So when the whole complex dCas9 plus the ADs gets guided to the right spot near a gene's promoter,
Guided by the RNA.
guided by the guide RNA, exactly, those ADs then wave over the cell's own natural transcription machinery. They basically force that targeted gene to be expressed, often really strongly, right there from its normal spot in the genome.
That makes sense. It's like putting a megaphone next to the gene's start button.
Kind of, yeah. A very targeted megaphone.
Which brings us neatly to optimizing this thing. Finding the best engine, right? The first version, dCas9 VP64, that was a proof of concept, made of 4 copies of VP16.
That was the pioneer, yeah. VP64. It showed that the basic idea worked,
Yeah.
But honestly, the activation was pretty modest.
Modest, okay? Not exactly groundbreaking for therapies yet.
Not really. For any serious clinical use or even complex research where you need strong, reliable gene activation, researchers knew they needed, well, more muscle. And that really pushed the field fast into developing and testing the next generation. And pretty quickly, some frontrunners emerged from those comparison studies, systems that really packed a punch.
Oh, yeah. 3 systems consistently stood out in testing as being way more potent. Those were VPR, SAM, and SunTag.
VPR, SAM, and SunTag. Got it.
And if you look closely at VPR, you can kind of see why it's so effective. Its design is pretty clever. It's a tripartite fusion.
Meaning 3 parts.
Exactly. 3 distinct, powerful activation domains all fused together. VP64, the original one, plus P65, and RITA.
A real power trio then. But okay, if VPR is showing these huge gains, you mentioned numbers like up to 320 times better activation than the old VP64.
Yeah, in tests on actual endogenous genes, it was a massive leap.
So why would researchers still bother with SAM or SunTag? Why isn't the most potent tool just automatically the winner?
That's a really critical point, actually. It's not always about just raw power. While VPR's design, that combo of 3 domains proved essential for maximum signal. Well, different cells, different genes, different goals might need different levels of activation.
Right.
Sometimes that sheer force of VPR, that huge 320-fold increase, it might actually be too much. It could stress the cell out, cause toxicity,
Okay, so you could overshoot the mark.
You could definitely overshoot. The key is finding the biologically relevant level of expression for what you're trying to achieve, not just cranking it up to 11 automatically. So having that variety of tools, SAM, SunTag, VPR, it gives researchers options.
That's really good context. Okay, now here's something else fascinating. Even with VPR, the most potent single activator, they found a way to boost the signal even more. This is where multiplexing comes in.
Correct. That was a bit of a surprise, actually. Even using the best individual activator, if you simultaneously targeted the same gene with multiple guide RNAs.
So hitting different spots near the gene's promoter.
Exactly, hitting multiple spots nearby, that significantly ramped up the gene induction level. It's like a stacking effect. And this strategy turns out to be super valuable for genes that are naturally tough to activate. Maybe they have really high baseline expression, the cell keeps locked down tight. Using multiple guides gives you a stronger, more reliable signal to overcome that resistance.
More guides, louder signal.
Oh,
Makes
yeah.
sense. But okay, anytime we talk CRISPR, especially for therapies, the elephant in the room is specificity, off-target effects. Are you accidentally turning on a bunch of other genes you didn't intend to?
Yeah, that's always the critical safety question, and it was looked at very carefully using RNA sequencing, looking genome-wide.
Oh, yeah.
The results were actually highly reassuring. For the vast majority of genes, expression levels didn't really change. The studies reported a really high correlation, like around 0.98 between control cells and the activator-treated cells for every gene except the one they were aiming for.
Wow, 0.98 is really tight correlation.
It is. So, for instance, when they targeted a gene called HBG1, that gene was massively upregulated, way more than anything else. It confirms these systems can be highly specific.
That specificity is just crucial, isn't it? Because once you have that kind of surgical control, you can start thinking beyond the lab bench and actually reprogramming cells, reprogramming life itself. Which takes us to part 2, CRISPR, in cell fate engineering.
Yeah, this is where things get really transformative for regenerative medicine. A major success story here is using CRISPR to generate induced pluripotent stem cells, iPSCs.
Right, making stem cells from regular adult cells like skin cells or even blood cells.
Exactly, taking easily accessible cells like human fibroblasts or these things called lymphoblastoid cell lines, LCLs, and turning back the clock to make them pluripotent.
So how did the CRISPR method compare to the standard way of doing this, the traditional transgenic methods using viruses and such?
Well, the big difference came down to quality and speed, really. Single-cell RNA sequencing gave a really detailed picture. What it showed was that the older transgenic methods, they often created a real mixed bag of cells, lots of cellular heterogeneity, not all cells properly reprogrammed, some going down weird paths,
Less consistent.
much less consistent. CRISPR reprogramming, though, seemed to guide the cells along a much more direct path to becoming pluripotent. You saw more uniform expression of the key pluripotency genes, less messiness, improved fidelity, and it happened faster.
So higher quality, more predictable stem cells, quicker. That sounds like a win for eventual clinical use.
Definitely a big step forward for consistency and reliability.
Now, getting this efficient reprogramming in human cells wasn't just about hitting the usual suspects, the famous Yamanaka factors like OCT4, SOX2.
Yeah, the canonical factor.
It needed something extra. The sources point out 2 other spots in the genome, non-pluripotency spots, that were like the secret ingredients. What were they?
Yeah, that was a key finding. It wasn't enough just to target OCT4, SOX2, KLF4, MYC, LIN28A. You needed those, plus targeting 2 other critical loci. One called the EEA motif, that stands for Embryo Genome Activation Enriched Allomotif.
Okay, EEA motif.
And the other was the MI302CHG transcript, which is related to the MIR 302367 cluster.
Let's dig into that EEA motif for a second, because the mechanism sounds wild. When dCas9 binds there, what's actually happening? It's not turning on a reprogramming gene directly, right?
That's what's so cool about it. Targeting the EEA motif didn't directly switch on, say, OCT4, but
Right.
hitting it was a massive boost to efficiency, like up to a 29-fold increase in reprogramming success.
Wow.
Mechanistically, it looks like when the dCas9 activator binds to this non-coding motif, it actually forces open the local chromatin structure. It like pries open that section of the genome.
So the tool isn't just acting as a gene activator here.
Wow.
It's using those activation domains to physically remodel the DNA packaging, like blasting open the door.
That's a great analogy. Yeah, the ATAC-seq data backs this up. It shows the chromatin opening up, and by clearing that space, essentially, it allows other key downstream pluripotency factors like NANOG and REX1 to get activated much earlier, about 2 days earlier in the colonies they observed.
That's fascinating. It's epigenetic engineering, clearing the path.
Exactly. It helps the cells follow that more direct, high-fidelity route we talked about.
Incredible control. And beyond making iPSCs, CRISPR is also letting scientists do direct conversion, right? Turning one cell type straight into another.
Yes, both in the dish, in vitro, and even starting to show promise in vivo inside a living organism.
What kinds of cells?
Well, for example, using VPR-based activation, researchers have successfully turned iPSCs into specialized nerve cells, iNeurons, by targeting key neurogenic genes like NGN2 and NurOD1.
Okay, neurons. What else?
And in the cardiac space, using multiplex CRISPR hitting multiple genes at once, specifically the GNT genes, GATA4, NK by 2.5, and TB by 5.
GNT. Got it.
They converted mouse fibroblasts, skin cells basically, directly into proliferative cardiac progenitor cells, or CPCs.
And you mentioned these iCPCs, these converted cardiac cells, might be safer.
That's a key potential advantage. They found these CPCs could be grown and expanded in the lab for over 20 passages, which is great for generating enough cells. But crucially, when they injected them into mice, they didn't form teratomas, those tumors you can sometimes get with pluripotent stem cells.
So potentially much safer for heart repair therapies down the line.
That's the hope, yes. And the flexibility even goes in vivo. There was a really neat demonstration using transgenic mice that were engineered to already express the dCas9 activator system.
So the tool is already present in the mouse's cells.
Exactly. And then by delivering just the guide RNAs, they could convert astrocytes, a type of support cell in the brain, directly into functional neurons right there in the mouse's brain. It shows this platform's potential for studying gene networks in a living system.
Wow, in vivo reprogramming, that's really something. And it leads us perfectly into our last part 3. Applications in disease research and high-throughput screening. Let's start with cancer, specifically drug resistance.
Right. A really important discovery here involves a type of ovarian cancer called high-grade serous carcinoma, HGSC. It's common and unfortunately quite aggressive. Researchers found that a known cancer-promoting gene, an oncogene called FOXM1, and another gene called RHNO1, are arranged in this really unique way on the chromosome. They're in what's called a bidirectional gene, BDG configuration.
Meaning they point away from each other but share.
They share a single promoter region between them, this FRBDP, and because they share that one switch, they tend to be turned on and off together. They're tightly co-expressed in the cancer cells.
So these two genes are kind of yoked together. Why does the cancer cell want both of them on at the same time? What do they do together?
Well, it turns out they cooperate to boost DNA repair, specifically homologous recombination or HR repair.
So they help the cancer cell fix damage caused by chemotherapy or other treatments, makes the cancer tougher.
Exactly. They work together to make the cancer more resilient. But this discovery also pointed towards a potential vulnerability. The researchers then tested the flip side of CRISPR using CRISPR interference, or CRISPR.
The off switch instead of the on switch, using dCas9 fused to a repressor.
Precisely. They directed CRISPR to that shared promoter, the FRBDP, to try and turn down both genes at once.
And did it work?
It did. Repressing that shared promoter successfully reduced the expression of both FOXM1 and RHNO1 simultaneously. And the crucial outcome was that this made the cancer cells, they used a line called OVCAR-A8, significantly more sensitive to a common cancer drug, the PARP inhibitor olaparib.
So disrupting that coordinated expression makes the cancer vulnerable again. That's a direct therapeutic insight.
It really is. It shows how understanding these control mechanisms via CRISPR tools can point to ways to overcome drug resistance.
Very cool. Okay, finally, let's talk about discovery using CRISPR. It seems perfect for these big, pooled gain-of-function genetic screens.
Absolutely. It's tailor-made for that. Instead of knocking genes out to see what breaks, you're systematically turning up potentially thousands of genes, one by one in different cells within a pool, to see which ones cause a specific effect or phenotype you're looking for.
Finding the genes whose upregulation matters.
Exactly. And a really timely example was its use in SARS-CoV-2 research during the pandemic.
For COVID-19. How was it used?
Well, researchers use these CRISPR screens to find host factors, our own proteins that influence viral infection. The screens correctly identified the known players, like the ACE2 receptor and the TMPRSS2 enzyme, which we knew were important for the virus getting into cells.
Okay, so it validated the approach. Did it find anything new?
It did. Crucially, these screens also uncovered a completely novel binding partner for the virus, a protein called LRRC15. That's leucine-rich repeat-containing protein 15.
LRRC15. And what was significant about finding that one?
Well, unlike ACE2, which helps the virus get in, LRRC15 actually seemed to inhibit viral infection. The thinking is it might act as some kind of decoy receptor, tying up the virus.
Interesting. So potentially protective.
Potentially. And what really underscored its importance was a finding from patient studies. Looking across the whole human proteome, the plasma levels of LRC15 showed the single strongest correlation with COVID-19 disease severity.
Wow. So high levels correlated with how sick people got.
Exactly. Which highlights how these unbiased activation screens can pull out really critical, maybe unexpected, players in disease pathways that you might miss otherwise.
Yeah, that's powerful discovery.
Yeah.
This whole deep dive, it really makes it clear that CRISPR isn't just, a small improvement. It feels like a really sophisticated, precise, and importantly, multiplexable platform for controlling our own genes. It seems like it's fundamentally changing how we can approach cell programming and even modeling diseases.
I agree. It's a paradigm shift in many ways. But, the challenge, as always, is optimization. We talked about how combining multiple guide RNAs boosts potency, which is often needed.
Right.
But researchers constantly have to wrestle with that delicate balance. You need enough activation to be biologically meaningful, but you don't want to push it so hard that you cause excessive stress or other problems in the cell. Like we said, the most potent tool isn't automatically the best tool for every single job.
Managing that dosage, that biological sweet spot,
Yeah,
is clearly going to be key moving forward.
absolutely.
Okay, so speaking of moving forward, CRISPR tech has already shown some really exciting promise in preclinical models, things like treating obesity, tackling retinal degradation by controlling key genes. So let's leave our listeners with this thought. The basic science of turning genes on seems increasingly well handled. The massive hurdle now seems to be delivery, right?
Delivery is always the challenge in genetic medicine, yeah.
So how does the scientific community crack that? How do we overcome the translational challenges? Things like the limited cargo space and safe delivery vehicles like AAV vectors, or manage the potential immune reactions our bodies might have to the bacterial dCas9 protein itself. How do we solve those to actually get this precise in vivo programming tool out of the lab and into the clinic?
That's the million-dollar question, isn't it? Solving that delivery puzzle safely and effectively is truly the next great frontier needed to unlock the full potential of CRISPR therapies.
Couldn't agree more. Well, that's our deep dive for today. Thanks so much for tuning in.